All right, here we go with our class, and this is where we're at for this week. We are going to continue to follow up on exam 2 and the answer key video, and learning as much as we can from that. And then we're talking about chapter 11, protein synthesis, and we have an assignment due this Thursday about protein synthesis, and it's covered in the chapter, but like our other assignments, I've tried to organize the material, and so the assignment covers really important parts of chapter 11, so it's a good, important assignment we want to get to. And in this case, there are just some short descriptions that you need to send me by email by Thursday. And then we have this RNA processing assignment that's due a week from Thursday. That'll be our last homework assignment of the semester. And it is not something that we're covering in the book. So RNA processing was dealt with in the book in chapter 6, and you don't need to look at that at all. And so that stands alone, but it's also a bunch of good information to add to our work, and it fits into the story of the nucleus because RNA processing occurs in the nucleus. So even though chapter 10, the nucleus that we covered for the third exam, doesn't have it, it's really important stuff that happens in the nucleus. So it's a good thing to put together like that. And then we have our third exam on the Thursday the 23rd, which is 2 weeks from this Thursday. And so that's going to cover 3 chapters. As it turns out, each of our exams have covered 3 chapters. Chapter 10 about the nucleus, and chapter 11 about protein synthesis, protein trafficking, and then chapter 14 about the cell membrane. So I put down the title of chapter 13, cell membrane, 14, because really early editions of this book that we're using actually had an extra chapter, and so it would fit in right about here. And so I don't know if it's possible that any of you have gotten that book. I just noticed it recently because I was looking at the publisher's website and seeing confusion. But it's just really clear, right? Our third exam is going to cover the stuff in the nucleus and about protein synthesis and protein trafficking around the cell, newly synthesized proteins, and then the cell membrane. So that's where we're at. Here's a look at our calendar where we're at, right? So we're here on Tuesday, and so we're going to be talking about more about protein synthesis and trafficking. We got an assignment due then, and then we have an assignment due there. We'll talk about the cell membrane. We'll easily get done with the chapter about protein trafficking and stuff like that Thursday. So we'll have a good amount of time to cover the cell membrane. It's a pretty straightforward chapter. We want to kind of connect it with the other stuff, the chapters and material we worked on. And then we have, after our third exam, here's our third exam, and after our third exam, we have just 2 more class meetings. We won't have any assignments, and maybe we'll have just a little bit of new reading after the third exam. It would not be a full chapter, it might be a part of a chapter. I haven't decided exactly what we're going to get to. And then our final exam is here on the 5th, like that. And our final exam covers the whole course. There'll be a little bit of extra new stuff, but it'll basically just be covering the whole course. But I want to point this out because I talked about it last week. So when you're gearing up to study for this, you're really also studying for our final. You try to pull it all together, and so it's worth it. If you look at the syllabus, each of the exams is worth more, a little bit more, a little bit more, a little bit more. And so here we have, lots of points. And that's hard, right? That's a challenging thing to do. There's a sort of a philosophy of grading that is new, a newer philosophy of grading, which is that lots of the points should come toward the end of the course, that the earlier points should be like not as much at stake, right? So I tried to do that with our first exam, where it's not as much at stake because the students are still going like, well, what is this? What's this course about? And so it's frustrating for students because I know, lots of points in all your courses. Just to add something here, I don't know if you do this, but the last week of classes, the instructors are not allowed to have any major assignments due. And if that's true in your class, you should just contact the dean and say, well, how are we going to deal with this? Because I'm not going to do it. Like that, right? And you'll get out of it. They'll talk to the instructor, and get out of it. So we're not going to have any major assignments due the last week of classes, okay? Like that, okay? This is just a thing from our syllabus. Yep, the final exam is on Tuesday. It's in this room. It actually starts at 8 a.m. But it's going to be the same style as our existing exams, and you'll have 2 hours to do it. And I'll mention this again. You don't 8 a.m. is earlier than 9:30, right? And so sometimes you're like, well, this is different. And if you get here, late, you don't have to get a speeding ticket because you'll have plenty of time to do it. And I don't have anything scheduled after 10. So if you got here at 9 a.m., you just get in here quietly and get your exam done, and we'll be in good shape. Okay, we'll talk about that more. So just to talk a little bit more about these 2 assignments. So the assignment that's due this Thursday is really laced in throughout this chapter 11. And so we want to talk some about chapter 11 today. And I'll talk a little bit about the assignment, but it's an assignment. It's covered in the assignment video. But just so we don't get too confused by trying to talk about the chapter before some of you have done the assignment, like that. And then again, the assignment that's due next week is not any part of any assigned reading. So that's really unusual. Most of the time we're covering the stuff an assignment as we're covering the book. But that next week, it's not any of that. You don't need to find the book to do the assignment or the learning for next week, okay? And so all of the stuff for exam 3 can be visualized, and that's good. We always want to try to do that and build that up, and we can walk through it. So the nucleus, chapter 10, protein synthesis stuff, chapter 11, and the cell membrane, chapter 14, like that, okay? And our assignments, so the RNA processing assignment that's due on Thursday here, it's in this. And then included in that assignment, I described this regulation, transcriptional, post-transcriptional regulation by RNA interference. That actually takes place in the cytoplasm. So that assignment for Thursday is really RNA processing inside the nucleus, and then this RNA interference outside of the nucleus in the cytoplasm. And then the protein synthesis stuff we're talking about this week, it's all stuff, that's a big story is in the cytoplasm. And we're especially interested in the regulation. And it would be post-transcriptional regulation, right? This is transcriptional regulation. That was the big part of our course. Then we have post-transcriptional regulation. And for protein synthesis, we could regulate the rate of translation and like that. But then we talk about post-translational regulation. And there are really 2 pieces to post-translational regulation, just to avoid getting people confused. There's the post-translational regulation that is right after the protein gets made. So the protein gets synthesized, and it's not ready. It's got to get processed, it's got to get adjusted and modified just to be able to do its job, like that. It's got to get to the right location and so forth. And then there's the protein floating around, just hanging out, ready to do their job, like FOXO. That FOXO that we learned about, that gets phosphorylated, that's just hanging out there. It could be FOXO that's, not new FOXO, it's just hanging out, and there's a pool of FOXO. And so that's what we are doing here. And then you'll see we'll start to talk a little bit about stuff that's got to get to the cell membrane, because the cell membrane is our next chapter. So these chapters all fold together into that image, and that image is really from Intro Bio. And so in Intro Bio, this is a figure from the Intro Bio textbook that's used at USM. It's used at USM for years. That's by this guy, Neil Campbell. And this just shows some eukaryotic organelles. So in that Intro Bio textbook, the first part of every Intro Bio textbook in America is about cell and molecular biology. So you jump right into it. There's an overview chapter about biology, welcome to biology, and then chapters 2, 3, 4 are chemistry. and introductory chemistry and water chemistry and covalent bonds and little molecules and bigger molecules like that. And all those chapters are heading to the cell, because that's what a molecular biology does, is he or she says, they say, let's just break the cell down to atoms and then put it back together and learn about the cell, the fundamental building block. So in Campbell, in other intro texts, this is like chapter 6 or 7, where they're like, now we're going to learn about the cell, and it'll be about eukaryotic cells. And so there's some talk about animal cells and some talk about plant cells, and they are trying to hurry along with so much intro-bio. They just put the organelles in a table. And we don't want to do that. We don't want to make it seem like, oh, it's just a table of organelles to memorize. We want to try to organize them into some stories that are memorable. And so we're talking about the nucleus and stuff that happens, in the nucleus, and we're ready for it because we got good examples of stuff that's got to get in the nucleus and out of the nucleus, So we're not thinking about the nucleus as just a table, a thing on a table. And then talking about the ribosome, and you'll see we'll talk a lot about the endoplasmic reticulum, the Golgi, the lysosome, and vesicles. And so, instead of just saying, oh, it's a table, we're going back to intro bio and fitting it into our understanding of processes at a molecular and cellular level like that, okay? And so when we talk about the endoplasmic reticulum, the AR, in our cells, plant and animal cells, eukaryotic cells, there's really two types of ER. And they are both named the same because they look very similar under an electron microscope. And so this is a transmission electron microscope, taking a picture, a micrograph, and a transmission electron microscope is TEM. So a TEM micrographs, and they're transmitted electrons, and so we see these organelles. And again, back in the 1950s, scientists would be like, oh, wow, look at those. I wonder what they do. And that's the birth of cell biology, and we're kind of talking about that a lot now. So rough ER and smooth ER, and they have very simple stories as to their main functions, and their main functions are very different. The rough ER is what we're talking about, especially because the rough part of the rough ER is little dots in these, little dots or bumps in these micrographs, and those are ribosomes. So the rough ER is the site of protein synthesis. And then the smooth ER is lots and lots of membrane. So it's like, pancakes of membrane, so lots and lots of membrane molecules. Lots and lots of membrane molecules, and it's that lipid bilayer, phospholipid bilayer, and the smooth ER is the site of hydrophobic molecule synthesis. So that's where all of the phospholipids get synthesized, is in the smooth ER, and then lots of other lipids like cholesterol. And lots of really important synthesis there. So fat storage molecules, stuff like that. And one thing that we see in these organelles and other organelles in eukaryotic cells is that something that was fascinating in the 1950s is that different cells have different amounts of these organelles. And so a fat cell, an adipose cell, has lots and lots of smooth ER, just lots of it, because it creates that hydrophobic spaces. And it's just the membrane where there's hydrophobic spaces. So we get into that a little bit. We get into a little bit in this chapter that's about protein synthesis and protein traffic. And the authors kind of got to tuck in the smooth ER because it's like, we got to keep these students from being confused, because they probably know about the smooth ER and the rough ER. And in chapter 11 about protein synthesis, it's all about rough ER. What's nice is we're going to go from chapter 11 to chapter 14. So from one phospholipid bilayer to another. In this case, the phospholipid bilayer of the smooth ER. Because we know, for example, that that we were interested in phosphatidylinositol, right? PIP. So it gets made in the smooth ER. Or phosphatidylserine on the inner leaflet of every cell membrane in our body, it gets made in the smooth ER. And so we're heading next to that chapter about the cell membrane. So this is where the cell membrane material gets made, like that. And for the rough ER, people like to talk about protein traffic. And they're talking about newly synthesized proteins getting to the proper destination. And we have lots of good examples of that. The PDGF receptor needs to be in the cell membrane. Newly synthesized PDGF receptor gets made here. And that's one of the examples in the homework assignment for Thursday. So protein traffic, and it's newly synthesized protein traffic coming through here. And then lots of these post-translational regulations for newly synthesized proteins happens in the rough ER and on their way to their destinations. On the other hand, totally different story, smooth ER. Newly synthesized lipids, and they're hydrophobic, and they need to get to their proper destination. So here, people talk about membrane traffic. And it's newly synthesized membrane. Let's move these molecules to their proper destinations. And in both cases, it's a challenge in the traffic, because in both cases, there's stories of where this needs to go and how is it going to get there. And so we want to talk about that in both cases. Okay, so that's the vision. Again, it's nothing like a table. It's like a real cell doing real stuff like that, okay? So here's just a picture drawing of the rough ER, and it's got a phospholipid membrane, and it's stacks of pancakes that are joined together, and it's rough because they're decorated with these ribosomes. And homework assignment talks about the ribosomes and how they get there. Inside of phospholipid-bound membrane, there's liquid. And inside of the membrane, in that liquidy environment, there's a general term, it's called the lumen. And so we want to just be able to use that term, the lumen, because we'll talk about the ER lumen, the lumen of the rough ER, and the lumen of the Golgi, and the lumen of vesicles, and the lumen of lysosomes, and so forth. So these organelles typically have liquid centers, yummy, and that liquid region is called the lumen, okay? So just some terminology there that we'll use going forward. And so just some figures from Thursday's homework assignment, just to clarify that. So this is a figure from chapter 11, and it shows the pathway of protein synthesis. So there's going to be these ribosomes that synthesize the proteins, and there's going to be an mRNA that's bound to the ribosome, and the mRNA is just guiding amino acid addition. So there are different locations the proteins need to go. And in the homework for Thursday, I say we can organize the big take-home messages from this figure and a lot of this chapter by talking about 4 proteins: hemoglobin, insulin, the PDGF receptor, and something called the lysosomal proton pump. And they fit into probably 90–95% of all proteins that need to get synthesized. Those 4 examples give us guides. And the first one is hemoglobin, and hemoglobin's just at our red blood cells. So again, genomic equivalents, all of our cells have all the genes. They all have the hemoglobin genes. It's only being expressed in precursors to red blood cells. And we know making new red blood cells all the time, from the bone marrow, and they're making a lot of ribosomes. And those ribosomes are just floating around in the cytoplasm. They don't have a special location they need to get to. They're just inside the red blood cells. And there are thousands and thousands of proteins that are just floating around inside the cytoplasm, like that. For example, that's where all the transcription factors are to start. They get made by ribosomes in the cytoplasm, and they're just floating around. All of the cytoskeleton, the microtubules and the actin microfilament proteins, lots and lots of enzymes just floating around in the cytoplasm. And those all get made by ribosomes that are also just floating around in the cytoplasm, and they're called free ribosomes. And I talk about this in the assignment too. They're made there, okay? And in general, those proteins that are in the cytoplasm, they're just totally made by ribosomes floating around. You know, the ribosome grabbed an mRNA and it makes hemoglobin, grabs another one and it makes foxyl, grabs another one, the ribosome just grabbing them and going like that, right? So that's great, and it talks about the destination of these proteins. And so it says, here's some proteins, and it doesn't even mention just that so many just get made in the cytoplasm, and it should, because there are so many proteins like hemoglobin that just get made in the cytoplasm. That mentions the nucleus. The proteins that get to the nucleus, we know that's a big deal. They get made in the cytoplasm, and they have to have a nuclear localization signal on them to get into the nucleus. So histone, DNA polymerase, and DNA repair enzyme just floating around in the cytoplasm. They get delivered after synthesis like that. And there's potential post-translational regulation, right? That you get sequestered in the cytoplasm, like FOXO or like CDC25. So it just lists that, and we got good examples in the nucleus. And then it says, oh, there's a lot of proteins that go into the mitochondria and chloroplast, and another organelle called peroxisome. Great. We don't go into those in detail. In general, those are just made by free ribosomes, and then they get delivered in some special ways, okay? So then there's a set of proteins like insulin that need to get out of the cell. So insulin gets made by cells in our pancreas, and it gets released into the blood. So secreted proteins. And there are a lot of proteins that need to go outside of the cell, like that. And so what about them? And so that's the second one in the homework assignment. And what happens when a protein like insulin gets synthesized is almost all the proteins that need to leave the cell are made the same way. And that is, they first start getting made by the free ribosomes. But those proteins, and again, it's really part of our homework assignment, so I'm just breezing through this to give us the context to talk about stuff here. Those proteins all have, at the first part of their protein sequence, they have a little portion called the signal sequence. And the signal sequence is giving the signal of, hey, we need to get out of the cell. And when a free ribosome starts to synthesize any protein that has a signal sequence, it says, hold on, I shouldn't let this go in the cytoplasm, it needs to get delivered. And so the signal sequence is the key for it to pause until it docks, they say docks, or binds to the rough ER. So all the ribosomes on the rough ER are actually free ribosomes that are synthesizing proteins like insulin and others that had to wait and dock. So that's the difference, right? A free ribosome is like a free-range chicken or whatever, a dog running around just doing its thing. But if they start synthesizing a protein like insulin, it needs to get released, they wait and they bind, and so the rough ER are all ribosomes that are just temporarily docked there, just temporarily parked, bound there as they finish synthesizing insulin, and they let go. And where does insulin show up? Newly synthesized insulin is in the lumen of the rough ER, all of it. And lots of other proteins are, and you'll see we want to talk a little bit more about that, but it's really dealt with in the homework assignment. And so it talks about that in this list. It's about secretions, secretory vesicles like that, okay? And then the third on our homework assignment is the PGF receptor, and we know, oh, it has to be in the cell membrane. It also, the PGF receptor protein also starts initially with a little signal sequence, so it starts getting the PGF receptor mRNA starts getting synthesized by free ribosomes, but then it's, whoop, you have a signal sequence, I'll wait until I bind here, then I'll finish synthesizing you. All the plasma membrane protein, almost all the plasma membrane proteins are synthesized in the rough ER. And so where's the location of newly synthesized PGF receptor? It's in the membrane of the rough ER. And then it's going to get delivered to the cell membrane. So I said, in our homework assignment for the 4 proteins, hemoglobin, free ribosomes, floating around the cytoplasm. Insulin, got to get out of this cell, has a signal sequence, the free ribosome binds and becomes a part of the rough ER until it finishes it, puts it in the lumen of the rough ER, then cell membrane proteins, signal sequence, rough ER, they get to be part of the membrane of the rough ER. And then the last one is called the lysosomal proton pump, and I have a little picture of that. So you probably remember what a lysosome is from intro bio. It is considered to be essentially the cell's stomach. You know that the inside of our stomachs are acidic, and that acidity helps digest food. And that's what the lysosome is. It's a membrane-bound vesicle that has a very low pH, very acidic pH. And it gets its acidic pH because in that membrane of the lysosome are a bunch of proteins that are proton pumps. They're pumping hydrogen ions in to create the acidic environment. So you gotta have them, right? They're a key concept for the lysosome is we need it to be acidic. How are we gonna do that? We're gonna pump hydrogen ions in, increase the hydrogen ion concentration. That's the definition of an acidic pH. And so that's called a lysosomal proton pump. And it needs to be in a membrane, but not the cell membrane. And so that's a story in protein traffic too, because the lysosomal proton pump, mRNA, binds initially to free ribosomes. And they're like, okay, I'll synthesize you. Oh, you have a signal sequence. I need to dock on the rough ER, and the location for newly synthesized lysosomal proton pump proteins is the membrane of the rough ER. And then that's gonna get delivered, and it's dealt with in the handout, to the lysosome. So good examples of proteins and protein traffic, where you gotta get. And it's about newly synthesized proteins, and we'll talk some more about that framework. The book talks about it, but the book doesn't give these simple examples where you're like, I got it. 4 examples are gonna cover it. Now, there are exceptions. You know, maybe 1 out of every 40, 50, 1 out of every 50 proteins doesn't take this pathway to get secreted. Doesn't take this pathway to get to the cell membrane. So there are exceptions to this, but they're unusual and interesting. But we won't mention them at all on our course because we're, introducing the main stories like that, right? And so that's part of the homework. I just go boom, boom, boom like that. And the homework is just to describe the pathway for each of them, each of the 4 like that, okay? And this is just a figure again from that, and it's just showing the signal sequence, and there's a signal recognition particle or protein that binds to it that causes a pause until it docks, and then you get, again, the lumen is this part. That's where any, with rare exceptions, any secretory protein first is synthesized. And the membrane, we can find almost all the cell membrane proteins there initially, almost all the membrane proteins for the lysosome. And if we just go back to this, our authors are trying to show you this. So it's got lysosomes on there, it's got plasma membrane on there, like that, okay? So we're not trying to explain all the examples, but good examples like that. And this, again, you don't have to scramble to write this down because it's the homework assignment, and for each step in the homework assignment, you just have to write a description of the pathway with that. And so the actual thing you'll send me is pretty straightforward. It just takes a while to explain it, okay? And so we want to go on from there, because that's a homework assignment, and if you haven't quite done it, we want to have some basic knowledge so we can get further along. And the basic knowledge continues. So here's the rough ER, and after these proteins are in the rough ER, they're going to get into the Golgi, and then they're going to get to the cell membrane or the lysosomal membrane. So that's protein traffic. And that's the story for the Golgi and the rough ER, is there are places where post-translational regulation is occurring on newly synthesized proteins. So a big category of post-translational regulation is just, let's get this protein ready. Let's do the step to get it ready. And so we'll talk today about some more examples, like insulin. When insulin is first synthesized in the lumen of the rough ER, it's not ready to be released from the cell. It needs to be modified. It needs to go through post-translational regulation to get ready to go, right? So there are these 2 large categories of post-translational regulation. One, just get it ready to do its job, finalize it. And for these proteins, it's happening in the rough ER, the Golgi, and then here some too, in these little vesicles. And that's how the material gets moved, the protein gets moved in these little droplets that are bound by phospholipid bilayer, and they're just called vesicles. And so that's what's shown here. These little droplets are these vesicles, and they're trafficking proteins to their proper destinations. Okay? All right? And so that's that. And so that's the big focus. But now we're going to say, yeah, we'll talk some more about that in class, and it's the homework assignment, and it's the major part of this chapter. But the author wants to tuck in, some features of the cell to help, put those in context. So that's a little bit confusing, but we're going to say, this is where we're going to talk about the smooth ER. And the smooth ER is not about protein synthesis and protein trafficking. It's about phospholipid and other lipid synthesis and membrane trafficking. Let's get these molecules to the right destination. And membrane trafficking, because the hydrophobic tails of phospholipid mean you can't just drag it through the cell. The cell is very hydrophobic. You have to keep it in its hydrophobic environment. And so we'll see with the smooth endoplasmic reticulum, it has the same strategy of pitching off these little droplets, these little vesicles, and carrying the newly synthesized lipids and fats to their proper destinations. Okay? And so this just shows it. It's tucked in here, and it's got nothing to do with protein synthesis because we're zoomed in on the smooth ER. And this is just showing the outer leaflet of the smooth ER, like that. So those are the phospholipids, and there would be another layer of them here, and then this would be the lumen of the smooth ER. I don't know, I'm sure there's stuff happening in the lumen of the smooth ER, but I don't think it's very much. Because there's synthesis happening here, and that means there's enzymes and chemical reactions, and the enzymes are hydrophilic, and so they're on this surface. And so this is where the chemical reactions are occurring to deal with lipids and fats, like that. So dealing with cholesterol. Cholesterol is the starting point for synthesizing testosterone and estrogen. And so if you look at our gonads, our testes or ovaries, and you look at the cells that make estrogen and testosterone, they're actually only a small number, a relatively small part of the testes and ovaries that are responsible for synthesizing those. And those cells have lots of smooth ER because, again, testosterone and estrogen are synthesized from cholesterol. Cholesterol is very hydrophobic. So the cholesterol would be hung up in here, and the enzymes would be here, grabbing at them and catalyzing chemical reactions. So that's what's going on, and that's the look of it. And you see how every time we have a phospholipid bilayer, there's two layers. And in the smooth ER, the action is just one layer. That layer that's contacting the cytoplasm where those enzymes could be, like that, and getting their job done. And so that's what it's showing here. See, it's showing, like that. And it's showing the synthesis, okay? So if you draw this out, we could do that, right? So let's just draw this idea out of the membrane traffic. So here we have some, this will be the smooth ER, and we'll say, these are the phospholipids like that, right? And the smooth ER is a phospholipid bilayer, so it's got two layers like this. And the activity is happening here on that brown layer because that's, that's facing the cytoplasm. And so it's this, and remember we like to use this word, it's the official word, it's leaflet. So there's an inner and outer leaflet like that, always with phospholipid bilayer, and they're usually really different. So if we just jump out here to the cell membrane, we would have, here's the cell membrane, and it has its outer leaflet, like that, in the cell membrane, and an inner leaflet, like that. Right? And we know, there's some important phospholipids that need to be in the inner membrane, like that, right? And so when these get made, they're going to pinch off, and then they will move into a vesicle. And so when we have a vesicle pinch off from the smooth ER, that vesicle will look like this. Okay, so it'll pinch off. It'll have that same orientation as the original smooth ER, like that. And so this is a vesicle, and they generally have pretty generic. Oh, it's a vesicle moving on its way. So it's got to pinch off and carry this, right? And so if you look, when this joins the cell membrane, we can draw where these will be. So essentially the outer leaflet of the smooth ER becomes the inner leaflet of the cell membrane, where that's where these phospholipids get added. And this process of joining it, just to add this, is when things happen like this at the cell membrane. This is a good term that you probably learned in intro-bio. That process of joining the vesicle to the membrane is called exocytosis. So we're delivering these vesicles to the cell membrane. We're adding newly synthesized phospholipids to the cell membrane, like that, okay? And so the newly synthesized phospholipids, they're always on the brown surface. And remember we said that you just never have those naturally switch to the other leaflet, because they have that hydrophilic head, and it's never going to go through the phospholipid. So in a sense, the smooth ER has no problem synthesizing phospholipids that are going to end up on the inner leaflet of the cell membrane. The trick is, how are we going to get newly synthesized phospholipids to be on the outer leaflet of the cell membrane? And we'll learn more in chapter 14, cell membrane. There's big differences between the outer leaflet and the inner leaflet, like that, right? And so, although this would never happen spontaneously to go from one side to the other, there's a lot. So we learned out here, right, there's a protein called scramblase, right, that is important in programmed cell death, apoptosis. So scramblase is going to do its job out here to move phospholipids. And we have a different family of proteins that flip, and they're called flipases. And there are flipases in the smooth ER. And they are a family of them, so they flip in different directions and so forth, but they're there to make sure we get the phospholipids on the leaflet where they're supposed to be. Are they supposed to be on the inside, inner leaflet of the cell membrane, or the outside? Like that. And so, does that run so that, the phospholipid with the programmed cell death, right, phosphatidylserine, it's on the inner leaflet. Great, you got made there. Do we need to flip it here? Nope. No, good, you're good to go. Get you out there, keep you on the inside, right? And then if the cell is going to go through apoptosis cascades to get activated, activate that scramblase, and it will flip them out there, like that. And it turns out that there are a bunch of proteins that do these jobs because they're pretty much phospholipid specific. Like, scramblase is going to flip phosphatidylserine. And that's true here. There's a family of these and other proteins. But we'll just learn that word flipase And we'll keep it simple and say we find flipases in the smooth ER. And their role is to flip newly synthesized phospholipids. If necessary, to say, oh, you're a phospholipid that needs to be on the outer leaflet of the cell membrane. Well, I'm going to flip you in the smooth ER. There's some of that flipping back and forth out here too. Flipase and scramblase are such great terms that kind of, they have names, like that's a protein, but they're also can be kind of the verb, in the sentence, like we're gonna flip you, we're gonna scramble you, like that. So it's unfortunate, and that's the reason why we don't want to dive into this story very much, because the naming of the proteins that do the switching of the phospholipids isn't like clean and satisfying. It's more like, oh gosh, kind of chaotic naming. But we got good names. Scramblase is in the cell membrane, right? Like that. And flipases are in the smooth ER. And so good information about the smooth ER, like that, okay? And that's the big role of it there. And so in doing that, see, the book wants to talk about that. It's just talking about those two leaflets, like that, right? Okay? And general term, membrane traffic. And every time, we talk about like those vesicles, they're involved in membrane traffic. So for the protein synthesis, we talk about the rough ER and the vesicles that carry newly synthesized protein from the rough ER to the Golgi. And then the Golgi is a stack of pancakes, and we have to move them from one pancake to the next, more vesicles. Then, after they're in the Golgi, vesicles carry the proteins either to the cell membrane or to the lysosome, maybe some other destinations, right? So anytime we're talking about that protein traffic, it's also membrane traffic. We're moving membrane material around like that. And so there's a significant relationship between the smooth ER and the rough ER, because the smooth ER is making all the parts of the membranes for the rough ER, the vesicles, the Golgi, and the cell membrane, right? So there's a lot of membrane traffic, a lot of molecules moving around. And again, you got to move them in a membrane because they have those hydrophobic tails, like that. So, that's the reason for all this traffic. And the cell is just busy with moving stuff around and moving these newly synthesized proteins and phospholipids and other fats around, like that. So protein traffic, newly synthesized proteins, again, with that, not to distinguish them, and not to confuse them. There's that protein flow, and just recognize that there are membranes involved there too. So there's just a big story, and membrane trafficking and the movement of lipid bilayers is a big part of cell biology, because so many of these organelles have a phospholipid bilayer around them, like that. Okay, so I have to have a little side note in this chapter about proteins, about the smooth ER. Smooth ER, not about protein synthesis. It's about phospholipid synthesis, lipid synthesis, fats and oil synthesis. Okay. So now we're going to go into this pathway of protein synthesis and talk some more about that pathway that looks like this, that we learn about with the 4 proteins in our homework that's due on Thursday. And our book wants to talk about this pathway some more, and so we're going to talk about it more. And it's like an add-on to the Thursday's homework. But you'll see, we're fine, even if you haven't done the homework yet, don't worry. I talked about it a little bit, right? And so it goes like this, right? So the free ribosome binds to an mRNA that needs to get to a specific destination, and it has a signal sequence. So that causes it to dock on the rough ER, and the protein gets synthesized in the rough ER. Then it gets moved to the Golgi, and then it moves to the cell membrane, or in our case, to the lysosome. So this is where there's a turn in the pathway for the lysosome. Stuff like insulin's going to the cell membrane to get secreted, stuff like PDGF receptors going to the cell membrane because it's part of the cell membrane. The lysosomal proton pump is heading toward the cell membrane. Right here, it needs to turn and go to the lysosome. And that lysosomal proton pump, that's kind of an important enzyme. And this pathway for the synthesis has other proteins that are on the pathway guiding that turn that it has to make. And it turns out there are some inherited human diseases that involve failure to properly target it to its destination. And when that happens, a person has lysosomes that don't get very acidic. And that person can be diagnosed with those syndromes by just looking at their cells and saying, wow, you have lots of lysosomes going on. And, why do you have so many? And the answer is they just can't get their job done. The garbage is piling up, right? The garbage workers are on strike, and the garbage is piling up in these people's cells, like that. So that pathway, right, is we're getting the protein to its destination. On the pathway, if it's, some regulation, like you got to turn here, there are other proteins that need to be in this spot to tell them to turn, and you can have mutations in those genes, and you don't get the traffic going to its destination properly. Sometimes the lysosomal proton pump ends up on the cell membrane in all of us by accident. And there are proteins on the cell membrane that are like, get back in there, right? Because it's just going to start pumping acidic stuff into the, protons into the cell. And we're making this all acidic, right? That's a bad move. So that becomes, the next thing to add is that to get to the proper destination, there are steps along the pathway, and those steps include proteins whose job it is just to guide them to the pathway. So we want to talk about some of that activity that's happening normally, because that can be messed up in human diseases, and you're like, whoa, you have problems with this because you're just not getting the protein to the right place. The lysosomal proton pump or the cell membrane proteins, they're good. You have wild type versions of those genes, but you have helpers along the way that you have mutant alleles for. So we're going to talk about those kind of ideas up. And this would be, again, post-translational regulation, but of this category of these are just newly synthesized proteins. We got to get them to their place. And so not the kind of post-translational regulation we've talked about a lot, where transcription factors are getting phosphorylated, and stuff like that. This is just the newly synthesized one. And there's going to be this post-translational regulating of them as they try to get to their proper destinations. And so we're going to talk about that, and we're going to talk about, again, from the book, try to pick some examples. And these would be examples of stories that, build on the examples from Thursday's homework assignment. So Thursday homework assignment, again, is just got to make hemoglobin, insulin, PDGF receptor, and the lysosomal proton pump. Got to get them to their proper destinations. So these would be examples of the post-translational changes that are occurring of these on these pathways, and they're not the same proteins. And I'm going to talk about insulin again, but they're not the same proteins. And you don't need to scramble to write them down, because we'll go through them one at a time, and we're going to talk about 4 examples. Again, right? So this is where we're at. We're on that pathway. This pathway, right? We're on this pathway. And these proteins need to be changed, regulated, altered on this pathway. And so places we'll talk about are what happens in the rough ER, it happens in the Golgi, it happens in these vesicles. So where does it happen? What's happening? Okay? And we just have these 4 examples. And our book doesn't try to organize it like this. Our book tells these stories. We got, figures from your book. Oh, snow day! I did not know that was going to happen. We should go outside. So the first one is proteins that are called antibodies. And we've talked some about antibodies. Antibodies are made in our immune system cells in the blood, and those antibodies get released. So they need to be secreted. They need to be released from the cell. So we're going to have a pathway for antibodies that's like insulin. The antibody genes get transcribed, right? RNA processing, they get out of the nucleus, they bind, the mRNA binds to a free ribosome, they start getting synthesized, and each of the ribosomal mRNAs has a little signal sequence that tells it you should make this in the rough ER. So it docks, that ribosome docks in the rough ER, the newly synthesized, or the site for newly synthesized antibodies is the lumen of the rough ER. They need to be secreted from the cell. So that's where they're going to be made and get done, okay? And I'll just show you a picture. And again, not a class in immunology. Don't need to know more about them than that. They're proteins that are going to get secreted from the cell. They get made in the rough ER, and their place is they're in the lumen, in that liquid center of the rough ER. So just to show you this, here's an antibody, and antibodies are made up of more than one protein chain, more than one polypeptide chain, and they get bound together to make the functional antibody, and they get bound together by these disulfide bonds. And so you learned about some of them in intro bio and probably in chemistry too. So disulfide bonds, two cysteine amino acid side chains have sulfur, and they join together. So these are covalent bonds holding these proteins together. And the antibody protein looks like that, where it's, you know, it's the functional protein is multi-subunit protein that's got to get held together. It's held together by these disulfide bonds, okay? And disulfide bonds in general get formed in the rough ER, like that. That's where they get put together, right? So they're ready to go, like that. We're going to see in just a short later example, insulin. Insulin is also held together by disulfide bonds. Where do you think those get made? In general, disulfide bonds get made in the rough ER. Okay? So that brings up this idea, and that is you've learned about proteins, that some proteins are held together by these disulfide bonds, disulfide linkages. It turns out, with almost no exceptions, proteins that get held together by disulfide bonds are outside of cells. Like secreted proteins, it's very common for them to have disulfide bonds. You know, and you almost certainly know about them, you know, from intro bio. Oh yeah, it's part of protein structures. You could have some disulfide bonds, great, like that. All those proteins we talked about in our course, all those transcription factors, none of those have disulfide bonds. And that's just a surprising thing, because it's one of the, you know, early things students learn about proteins is, oh, you can have a important part of the protein folding can be forming these disulfide bonds. They really help hold things together and hold the proper conformation. Yep. So we just want to talk about that, and we want to do it this way. We're going to get to this image and say, where's that image? That, put it here, the lumen, so draw a little arrow to this, the lumen of the rough ER, the rough ER lumen, so that liquid interior is an oxidizing environment. The cytoplasm, and in general the inside of the cell, outside of the lumen of the rough ER and outside of some other locations, the cytoplasm is a reducing environment. And the relevance for that here is that a cysteine amino acid has this sulfur group, and in a reducing environment, the sulfur is reduced, and you have it as an SH, and in an oxidizing environment, the SH is oxidized, and we get the disulfide bond form. So the bonds form because of the environment, the chemistry of the lumen in the rough ER. And it's an oxidizing environment, you know, and these proteins have to go through the Golgi. The Golgi also has an oxidizing environment, and these vesicles heading out. And then the extracellular environment, you know, so our blood and so forth is considered an oxidizing environment. And so that's a big distinction in post-translational regulation. This is a covalent change to the protein, so this would be a post-translational regulation that is a post-translational modification. And the modification is, well, do you have disulfide linkages, or do you just have sulfhydryl groups? And yeah, it's just a huge difference in the story of protein structure in the cytoplasm and, you know, generally inside the cell versus outside the cell. Yep, okay, with that. So that's our first example is disulfide bonds in newly synthesized proteins. Disulfide bonds form in the rough ER. Disulfide bonds formed in the rough ER. Our example is antibodies, but we'll see insulin also fits into that story. So lots and lots of proteins that leave the cell have disulfide bonds. Let's just use this example here too. We could talk about this. So, you know, when we look at this, we can have this come up. Let's just say, here's the cell membrane. And we have these transmembrane proteins, like, for example, PDGF receptor, right? So we have a lot of these receptors. So for any of these receptors, right, if you said, do they have disulfide bonds, disulfide linkages? The answer is not in this part. No disulfide here. Outside, maybe. Got to study them and find out. That, I don't know why my pen is not working up that high. So maybe outside. That is just the case of those proteins, that they can have a big role in a receptor, transmembrane protein, on the outside of the cell to have disulfide bonds help fold the protein, hold it together, but not on the inside. Yep, okay. So that's our first example, the antibodies doing that, okay? All right, so that's an example of post-translational modification, covalent change to a protein, and it happens in the rough ER. Like that, okay? And it's just saying that again, like that, okay? So our second one is, the modification is called glycosylation, and it happens in the Golgi. And our example is that lysosomal proton pump. And so glycosylation is the covalent modification of proteins where long chains of polysaccharides get covalently bonded to the protein. And you might have learned about that a little bit before, but the result is a glycoprotein, and so let's just draw that out. Oh, we just add to it here. So for some of our receptors that we've talked about, they actually have glycosylation, and the glycosylation would be here. So that would be a polysaccharide made up of long chains of monosaccharide sugars, polysaccharide, and this was added by this glycosylation, which occurred in the Golgi. And that's that idea of modifying newly synthesized proteins. That's really the function of the Golgi. So the rough ER is a site of protein synthesis, and the Golgi is a site of protein modification by glycosylation and other ways. So why do we have to go through the Golgi? Because we need to do some post-translational regulation, including glycosylation. And there too, right, we talked about this just a minute ago with the disulfide bonds. Glycosylation is really a story for proteins that are going to be outside of the cell, either secreted or membrane bound with a part of the protein outside the cell. And so the cytoplasm, these modifications that happen in the Golgi, none of the Golgi modifications really are happening in here, in just the free, cytoplasm of the cell. And so let's talk about that a little bit more. So here's an example. The book is showing this in a figure. And the book shows actually quite a bit of this. So glycosylation is actually important for these proteins that are outside the cell in a lot of different ways. And so the book wants to talk about it a lot. And we're just going to focus on one example of glycosylation. So these are sugar monomers. It could be like glucose or fructose and so forth. There are lots and lots of different sugars, ribose and so forth like that, right? And so there's some, you're like, I never heard of that. It's a sugar. And the sugar we're going to learn the name of is mannose. So, in the modification in the Golgi, like that, some glycosylation occurs, some change of polysaccharides occur, and that happens to the lysosomal proton pump. So remember, we learned about that. That's got to make its way to the lysosome, right? And it must have a tag, right? There must be some sort of label on it to go there, right? And remember I said that turn happens after the Golgi. It's in the Golgi where they get tagged, and the tag is this. It's a mannose, and it gets phosphorylated. So the tag for the lysosomal proton pump is mannose 6-phosphate. So there are enzymes in the Golgi, and these enzymes are proteins, and they need to tag the lysosomal proton pump so that it will go to get carried to the lysosome. And so these would be examples of enzymes that have genes. If they're mutated, the person has this, and they're called lysosomal storage diseases. The person does not have good lysosomes. They got lysosomes, but they're not very acidic. So they're storing the garbage. They're not, breaking it down like that. Yeah? And so that's happening, and this would be in the lumen of the Golgi. Like that. So in the lumen of the Golgi is where these enzymes are, these polysaccharides are being added. Like that. And our favorite one is the lysosomal proton pump, but there are many proteins. Remember, those proteins they're leaving the cell, or they're going to be on the cell surface, they're going to be outside of the cell. Here's an example where they're going to stay inside the cell, not going to be floating around in the cytoplasm. The mannose 6-phosphate is going to be actually pointed to the inside of the lysosome, just because of its orientation and position. So it gets tagged there. And so that's our example of a modification of a glycosylation. It's a great one because it's going to help just get it to its destination. Proteins get glycosylated and go onto the cell surface or out of the cell for a whole bunch of different reasons. Ours would just have one, and that is, well, you got to glycosylate this to get it to target to the lysosome. And then let me just show you this again, where's that figure where it shows. So this is zoomed in, our book shows this, and this is zoomed in, that's the Golgi, so the rough ER is here, there's the rough ER coming to the Golgi, getting glycosylated in the lumen of the Golgi, and they're coming out here, and they're vesicles that will be heading to the cell membrane to exocytose and release lysosome or antibodies and other proteins secreted, or join the membrane by exocytosis and bring the cell membrane proteins, the transmembrane proteins to the cell surface like that. So it's in these vesicles where there's more stuff going on, like traffic directions, This is where the lysosomal proton pump is recognized as having mannose 6-phosphate on it, and it's like, go this way, don't go to the cell membrane. And just to distinguish that, these vesicles here are called endosomes. So we should put that word up here. It's just a vesicle, but it's just kind of a specialized setting. And so scientists would be like, there are scientists that spend their career on these vesicles because they do take a turn here to go to the lysosome, and so the scientists like to call this type of vesicle an endosome. So it's good for us to see that. It's in the endosome where the lysosomal proton pump gets turned to go to the lysosome. Newly synthesized lysosomal proton pumps, they start out in the rough ER, and they're in the membrane of the rough ER, then they're carried to the Golgi through these vesicles. Then in the Golgi, they get glycosylated. In their case, they get a specific kind of polysaccharide that's got mannose. It's got mannose 6-phosphate on it. So when they get to this vesicle here, the vesicle's like, oh, okay, all right, you gotta take a turn here. We do not want you on the cell membrane. It's an important turn. We need to get you over here to the lysosome. So sweet, really sweet cell biology stuff, right? You could have a one-act play, right? And all the characters are, you know, Don Diego is going to be the lysosomal pump, you know, and Chase is going to be insulin, and Naomi is going to be antibodies like that, and you just have them all going through the stages and being like, oh, you know, Don Diego has to get a, you know, label on his shirt that says, you know, mannose 6-phosphate, and then, you know, Thomas is going to be like, okay, I'm in an endosome, I'm just hanging out in the endosome, and I'm just guiding you over to the lysosome, right? So again, it's this thing of just draw it out, make it big. There's so much of our learning for the third exam that's draw it out, make it big. In this case, because it's cell biology, we've zoomed out a little bit. You know, our assignments early in the course were molecular biology, and we're zoomed in, you know, and we're looking at the little molecules and how the, you know, MyoD binds to the enhancer, right? We've zoomed out a little bit, but it's still the same thing. It's very physical and very sensible, like that. Yep. Okay, so that's our second example, and it's glycosylation, and our favorite glycosylation story is we gotta glycosylate the lysosomal proton pump. I didn't write this word down. Proteins that are glycosylated are called glycoproteins. So the glycoproteins, and there are many of them. In general, the glycoproteins are on the cell membrane on the outside, or they're secreted, or they might be specialized ones that have to go to a place like the lysosome. And so the lysosomal proton pump is a glycoprotein. Fabulous, okay? So then the next one is a post-translational regulation step where the protein needs to get cut. And so we've learned about some proteins that get cut. And when a protein gets cut, the enzyme that cuts it is an enzyme called a protease. So we gotta have a protease. And in life, there are really two categories of proteases. There are proteases that are digestive enzymes. And their job is to break proteins down that we eat and break them down to individual amino acids. Our individual cells have to do that too. They'll have proteins that are like, okay, we need to break you down. So we're gonna break you down, we're just gonna cut you up like that. So there are some proteases that are just general scissors, and they're just clip, 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 like that. Then there are other proteases that have very specific targets, and they cut that protein like that. And they're not destroying it. In some cases, they're activating it, because it needs to get cut to be activated. And so there are proteases that need to cut insulin, and so they must be on the pathway before insulin leaves the cell. So this would be getting insulin ready to go out of the cell. That's our example there. All right, and so here's a term like that. Insulin has a funny name when it's newly synthesized. Insulin is called a pre-pro-insulin because it's synthesized as a longer molecule, and it's going to get cut. And it's a pre-pro-insulin because there are two cuts that have to get made. I've mentioned that the insulin has to have a signal sequence on it that shows it, the ribosome, the free ribosome, you know, you're making a protein that needs to be made of the rough ER. That signal sequence that's on insulin, that's going to get cut off. And when it gets to the rough ER, it gets cut off. It's just an extra piece, you know, it's a tag, a flag to get it there. So that's the pre part. And then the pro part is that when we look at the structure of insulin, this is the pro-insulin. So pre-insulin had a little bit more signal sequence on it. The pre part got cut off. Now it's pro-insulin. It's got to get cut by some different proteases because this is what the mature insulin molecule looks like when it's floating around in our blood. Okay? And that's just always the case. And so that's got to happen in the cells in the pancreas. And where in the cells? Well, in this case, it says that the pre-segment, signal peptide, directs the endoplasmic reticulum, where it's cleaved to proinsulin, which is then processed to mature insulin. And so that happens in the lumen of the rough ER. So as the free ribosome binds the rough ER and says, I'm bringing a protein, I'm going to make it in the rough ER, the little pre-piece gets cut off. That's the signal sequence that was needed to get it there. And then after the insulin gets made, in the rough ER, there is a protease that cuts it to make the final insulin. And so see how the proinsulin is pretty long protein, and then part of it gets cut and just separate. So this is the final insulin that would be in our blood. And you can see that before it gets cut, disulfide bonds are added. So I mentioned that that's where it happens, in the rough ER. The rough ER lumen is an oxidizing environment. Proteins that have cysteines want to form disulfide bonds. That happens in the rough ER to the proinsulin protein, and then it gets clipped by a protease. The protease is a protein. I don't know the name of it. We don't need to know the name of it. Well, how did it get to the lumen of the rough ER? That protein, the protease enzyme, it must have had a signal sequence. So when it gets synthesized, free ribosome says, oh, I got to get you to the rough ER, because you're going to spend your time in the lumen cutting insulin, helping get ready, like that. Yep. Okay? Like that. Yeah. And just to help this be memorable, this is a funny fact. This part looks like it gets thrown away. This part is actually a hormone called C-peptide. And see how you're making it in equal amounts? There's a thing where, like, insulin gets used up in our blood. And so when a doctor is first testing a person to see, like, what's the deal with your insulin, they measure the C-peptide, because the C-peptide is more stable. And so they've known about it for some time, but they just thought it was the C-peptide and kind of this extra piece. And now they see, oh no, it's actually got its function on its own, like that. So just to help you remember, right, that the insulin's got to get processed, it's got to get clipped, just remember that, oh wow, not only do we get insulin out of it, we get this other piece that is the insulin, but it's not really, it's because it's not the regular part of insulin, it's just this extra piece, but it has its own different job to do out in the blood. So both of those guys are going to go through and get secreted. Yep. So that's our example about that. This processing, the cutting with proteases, that happens generally in the lumen of the rough ER. Okay, and I just was reminded of this. Just a little bit about the C-peptide. That's not our focus, but if you're interested in insulin and human health, it is fascinating to see. Oh, wow, there you get another hormone out of the deal, like that, and scientists, study it as part of diabetes and so forth, like that. Yep. All right. And so, what is that right there? I don't know. So then our last one, and we got to get out of here. We have to get out of here. We'll deal with our last one on Thursday, but it's RAS. Yay. So familiar protein. So we're just going to have a list of 4. Our book talks about the stories, but doesn't use a nice, crisp example, like, set of examples to get us there. Okay, so that's where we're at. Remember, you have the homework assignment due on Thursday, and then the last homework assignment we'll have for our course is due a week from Thursday.